Hello everyone, my name is Giovanna, and the title of my study is Prostate Development of Females Gerbils. The presence of a female prostate gland in some mammal species, like in rodents, bats, and rabbits, is already known. Nonetheless, there's still lack of information regarding its biological features, especially in its development. So, the aim of this study was to characterize the initial events of prostate development in female gerbils. Here we have the histology of the urogenital sinus of a one-day-old female gerbil. And here we have circled by these dots the epithelial buds that will interact with the periurethral mesenchyme cells, and this interaction will allow the bud cells to turn into the acini of an adult prostate gland. To perform this study, we used the offspring of 30 pregnant females and divided them into 6 experimental groups, as we can see here. The letter E represents embryonic day and the letter P represents postnatal day. The urogenital sinus of all fetuses and newborns were collected and underwent the following procedures. Immunofluorescence for alpha-actin, immunohistochemistry for androgen and estrogen receptors, and for p63 protein, 3D reconstruction, and morphological analysis. Here we have the 3D reconstruction, and we can see in blue the urogenital sinus epithelium, in yellow the epithelial buds, in red we can see the smooth muscle layer, and in a lighter color blue, we can see the periurethral mesenchyme. So we can see that the initiation of the budding process begins at the 21st embryonic day. And at the 24th embryonic day, we can see that only one bud of all that emerged by the 21st embryonic day reached the periurethral mesenchyme. And we can see here that this bud had to cross the gap that exists in the smooth muscle layer to reach the periurethral mesenchyme. And curiously, the orange of this bud that reached the periurethral mesenchyme is far from the bladder neck, that is a region located here. And in the postnatal day 1, we can see that this bud is entirely reached the periurethral mesenchyme. Regarding the immunohistochemistry for p63, we can see that by the 20th embryonic day until the 22nd embryonic day, the p63 positive cells are in a multi-layer distribution, as we can see here in the UGE and also in the buds. But from the 23rd embryonic day, these cells became restricted to a basal compartment, constituting a monolayer of p63 positive cells in the UGE and also in the buds. As for immunohistochemistry for androgen receptor and for estrogen receptor, we can see that the urogenital sinus mesenchyme cells were expressed in both of these receptors, but we can observe a stronger staining in the smooth muscle layer for the estrogen receptor, and in the periurethral mesenchyme we can observe a stronger staining for the androgen receptor. From these results, we can conclude that the periurethral mesenchyme seems not to be required for budding induction, but it is necessary for the upcoming phases of prostate development. ER alpha and AR may be involved with paracrine pathways that are important for prostatic development. And we can also conclude that the UGE cells became committed with a epithelial phenotype in the early phases of prostate development, once these cells undergo to a fast differentiation process. Thank you for the attention.